guys, thanks for joining us today. I'm Mary. I'm coming to you live from the Pro Museum, and I'm so excited that we have the opportunity to present some live webinars to you today. If you see me glance over at my computer, I'm just checking things out and making sure that everything is rolling well. We have put the chat window up, so you're more than welcome to throw in questions as we go through, and I'll answer those as they come across the screen. And if you're on a cell phone, you can access that chat window at the bottom of your screen. It'll say Top Chat. You just click there and it'll bring that up on a cell phone or a tablet. And if you're on your computer, it'll be on the right side of the screen where you're viewing. All right, so today's topic that we're going to be going over is paper chromatography. And this is a subject that we see used a lot in chemistry classrooms or in biology classrooms, but it's something that you can do at all levels. And so we wanted to introduce you to an activity that would be great to do, especially if you have testing right now and you're looking for something those last few weeks of school. So just to start with, I want to talk to you a little bit about what chromatography is and how we can use that in the classroom. So first up, paper chromatography is a technique where we separate pigments of different substances or chemicals. And once we separate those substances, then researchers can study and analyze those components. And so there are lots of different types of chromatography. We have liquid chromatography, there's gas chromatography. Today we're going to be doing liquid chromatography, and we're going to be doing that using just water. So everything we're doing today is perfectly safe to do in your classroom, and it's all things that you're going to have access to easily from Walmart or Target or whatever your other big box store is. And when I mention store names, this is not sponsored. We're not advocating for a certain store, just giving you some ideas on where you can get these supplies. So we are going to, as I said, look at paper chromatography today using liquids. And as technology has improved, so has chromatography. And in fact, today, most chromatography is done using machines. And so we use it for things like blood sample and analysis. And so you would just put your sample into the machine. It would go through one of the various chemical processes to do a chromatogram, which, because of those advances in technology, has really left paper chromatography as something that we see just in the classroom. But it's still a very useful process that you can take your kids through, and you can still discover a lot about science using this process. So, like I said, everything we're doing today is perfectly safe. We're going to be dealing with just standard school supplies. So what I have here on my table is just a beaker full of water, and a clothespin and a popsicle stick. We have a couple of empty beakers, or you can even do this using cups, ruler, pencil, scissors. We're going to use coffee filters, which I have one that's done here, but these are just standard coffee filters, again, that you can buy at a big box store, and I've just kind of stored them flat so that they're easy to use for this process. And then over here on the other side of the table, I have some markers, and I'll talk to you more about those when we get to that point in our process. So first I want to walk you through a little bit of the vocabulary that's involved in paper chromatography. Some of this is vocabulary that students in the classroom will need to know for whatever test they're taking, and it's typically vocabulary that's associated with chemistry. But then we also have some chromatography specific vocabulary. Sorry about that, I'm getting my words all twisted up here. So I've written some of these vocab words over here on the board, and we're just going to go over and make sure that we're all on the same page with understanding as to what these are. Let me adjust my screen here. Make sure we can see the board well. Perfect. All right, so one of the first words that we need to make sure we all understand is the word solvent. And again, solvent is one of these chemistry words that's typically in most curriculum. And solvents, as we know, are just liquids that are dissolving other substances. And if you're looking at an entire solution, the solvent is going to be the highest amount, the substance that's in the highest amount within that solution. Then we also have the word solute, And solutes are typically the compound or the chemical that's dissolved inside of a solution. And so that's going to be the lesser amount of whatever you find in a mixture. And then we have solution, and solution is a mixture that's combined. So you have the solvent dissolving the solute, and both of these things together are the solution. And we're going to refer to the solution today as a mixture. So we'll both talk about mixture and solution. And then we have this term capillary action, and this is something that we sometimes introduce in upper middle school or even high school. But capillary action is when water flows through basically narrow tubes, and it can flow both with or against gravity. But the neat thing about capillary action is you don't have to have gravity, and so water can actually travel against gravity in an upwards or sideways fashion. And the whole idea of chromatography is based in this principle of capillary action. So it's a great way to talk about one of the properties of water. Then the words that you see in green, these are specific to chromatography. So the first word is called mobile phase. And in chromatography, the mobile phase is what is actually moving through whatever solid you're working with. So this is going to be when we're looking at the solvent carrying all of the solutes up through the paper that we're going to be using. The stationary phase, stationary just means not moving, standing still. So in our case today, stationary phase is going to be the solid that's not moving, and our solid is going to be the paper that we're using. And then the last word is called the analyte. And in chromatography, the analyte is just the solution or the mixture that has the compounds you're trying to analyze. So we've already said we have a solution or mixture. Analyte's just another version of that. Hi, Aparna. Glad you were able to join us today. I see that you've said hi in the chat, so welcome. All right, so that's a little bit about the vocabulary for chromatography. And so today, our chromatography paper, or the word we learned over here, our stationary phase, is going to be the coffee filter. And as you can see here, I have a little square piece of paper, and these are really easy to create. So this larger filter can get a little bit bulky. So what I've done is I've just used a pair of scissors and I've just trimmed a little square out of the middle of that coffee filter. And that's it. And then our mobile phase today, we're going to be using water as our solvent, and then we're going to be using markers as our solute. And so I just have some various brands of markers. So I have the name brand, and then I have just as kind of an off brand, and then I have chart markers, and I have them all in the same colors so that we can compare the difference between them. All right, so let's get on to the exciting part, the how does chromatography work? What are we going to do? What are we going to set up here? So I have a drawing over here on the side of the room. Let me zoom out so you can see the whole field of view here. So these diagrams are just a way for you, let me zoom in so you can see those a little bit better. There we go. That should be in the field of view now. And so here's our chromatography paper. That's the piece of paper that I was just showing you that I cut out from the coffee filter. So there. And then what you're going to do is you're going to draw a pencil line about 1 to 2 centimeters from the bottom of that paper. And then you're going to put your mixture sample on the line you drew. Now it's very, very important that you use a pencil, because if you use a marker or a pen, the whole idea is we're trying to separate what's inside of that marker dot in that mixture. We're trying to separate out those compounds. So you want to make sure you use a pencil line. All right, so to do chromatography, the way this works is that we're going to separate out the compounds that are inside one single solution, one mixture. And so to do that, we're going to place a sample of that mixture, like I said, on the chromatography paper. So I'm going to start by drawing a line about 1 centimeter from the bottom of my chromatography paper. Let me slide this tray out of the way so I can use the table here. And I apologize, this is the farthest that I can zoom in on this camera. So there are instruction sheets online for this, and then I'll also be posting an instruction guide for this on Edmodo, so you can access that later. So I've measured up 1 centimeter from the bottom of my paper. I'm going to draw a pencil line there. And I know you probably can't see that pencil line super well. Let me see if I can get it down here on the table. It just doesn't want to show up, does it? Put my hand behind it. I can kind of see it. So that pencil line runs from here to here. All right, so in order to separate out those pigments, we have to put the pigments on the paper. So to do that, I'm going to set a sheet of paper behind this just so I don't mark on my table. Let me put blue behind it. That way maybe you can see what I'm doing a little bit better here. All right. So here's my paper. And what I'm going to do is I'm going to place a dot of each of the markers that I have on that line. So here's my name brand. I'm just going to put a little dot of name brand green. And you want to saturate the paper with that. So it should, if you've done it correctly, soak through to the back side. Then I'm going to do the same thing with my off brand. And just from visual observation, this one seems to be a bit more liquidy. It put the pigment on the paper a lot better, and then the pigment's already spread out quite a bit. So it seems like the name brand is a drier marker. I mean, I don't know if that's because of the age of the markers or if that's just how they're made. And then last but not least, I'm going to put a dot from a chart marker on here. And that one seems to be the darkest pigment of all. So that'll be interesting to see what happens when we separate these out. All right. So now that we've got our samples on our chromatography paper, then we're going to fill a small container with some of our solvent. Now for different types of chromatography, we have different solvents. So like I said earlier, today we're just going to use water. Some experiments will call for alcohol, like isopropyl alcohol, rubbing alcohol. Some experiments use vinegar. I've used vinegar for this experiment as well, and it works quite well. And then some experiments will actually call for specific chromatography solutions based on the compounds you want to separate. For example, the plant pigment chromatography that we do a lot in the high school level actually uses a petroleum ether substance. It's pretty volatile, not something that you want to just use in any situation. You definitely want to use the solvent that's going to be the least dangerous for whatever you're doing, because it's always about safety first. So we're just going with water today. So then I'm going to take my beaker and I am going to pour just a little bit of water into the bottom. And I'm using distilled water. All right, so let me zoom in so you can see that. Apologize for not zooming in there. All right, so now that we've got that water in the bottom, I'm going to hold my piece of paper right up to that beaker. And I'm going to make sure that just the bottom edge of that paper is going to get into that beaker. So the idea here is that you want your solvent to touch the bottom of your filter paper, but you don't want it to touch your samples because that can cause them to leach back out into the water. So it looks like we're about right here. The other thing is when I put this paper into the beaker and I let it touch the water, I want to make sure that the paper, as best I can, doesn't touch any of the sides of the beakers and it's just hanging there suspended. So sometimes if your paper is long enough, you can make a cylinder out of it and just set the cylinder in there. We don't have that luxury with the way I'm doing it today. Let me turn this around so you can see what's happening. So instead, I'm just going to rig up a little suspension system. So I'm going to take my popsicle stick and I'm going to lay the very top edge of my chromatography paper onto that popsicle stick. And then I'm going to fold it down so that the popsicle stick is flat and the paper's hanging off the edge. And then to hold that on there, you could use a piece of tape, but sometimes the tape doesn't work because eventually this is going to get wet. So I just like to use a standard clothespin, and I'm going to clothespin, kind of at a weird angle, but it works, the piece of paper onto that popsicle stick. And there you go. All right. So once we have the paper like suspended and ready to go, and we're ready to separate out those pigments, then what I want to do is create a sealed system in this jar. The idea is that you want that water, that solvent in this case, to wick up the paper by capillary action. So it's going to go up the paper, and it's going to pick up any of the dissolved compounds that are inside that little mixture, that compound substance that you put on your line. Now, in order to do this, we need to make sure that the paper stays wet throughout the entire process. If the paper dries out, then it's going to stop the movement of those compounds, the separation as they move up the paper. So we want to seal in the water vapor into this system as best we can. So if I were to just stick this piece of paper in there like it is now and start running the chromatograph, it would work. But sometimes, especially if you're using something like acetone or alcohol, that paper's going to have the moisture and that solvent evaporating before it has a chance to pick up the compounds. So we're going to create a closed system by putting a cover over the top of this beaker. Now there's lots of ways you could do this. You could put some plastic wrap over it. I've used, one year when I was teaching, I used plastic shower caps like from hotels that I just like hooked over the top. I've put plastic shoe boxes over it. There's all sorts of different methods that you can do. Some places have beakers that have lids. What I found works really, really well, let me reach over here, is a bigger beaker. You can just invert the bigger beaker over the small one, and you've created a closed system. Yeah, a little bit of escape will happen here at the bottom. You can seal that off, but it's usually not that big a deal for what we're doing. So now that we know that we're going to have a sealed off system, and we're going to make sure that we get full separation of our pigments because we're not letting any vapor escape, we're going to check our rigging here. My clothespin has slipped just a little bit. There we go. We're going to drop very carefully our piece of chromatography paper into the solution. We're going to invert our larger beaker over the top, making sure we don't let anything touch. Yeah, we're good. So nothing's touching the side. Let me zoom in so you can see that a little bit better. Okay, so we're running that chromatograph. Now it's going to take a minute for it to run, and you can probably already start to see a little bit right here that's where the solvent front is, and so it's starting to separate, and it's moved about an inch at this point. So we're getting close. Now the idea is that you want to stop the chromatograph before it reaches the top for two reasons. Number one, there is a measurement that you can take that we'll talk about in a minute. And that measurement, you don't want to go all the way to the end of your paper, because what can happen is that compound can be carried all the way to the end, and then you don't get a good measurement. The other reason is that you want to be able to see the separation, and you want that to stay towards the bottom. So we're going to stop this about a centimeter or so from the top. I'll probably stop at about 2 because I found the water keeps wicking about 2 centimeters when you pull these out of solution. Okay. Now when we look at the piece of paper, the wet edge that's on the top, that's called the solvent front. You can think about it as just being the end of the solvent, or how far the solvent has gotten to. And so when I was saying we want to stop before the solvent reaches the edge, we want to look at that solvent front and stop about 2 centimeters from the top. It looks like we have about 4 more centimeters or so to go. So while we do that, let's go back over here to the chart. We'll let that keep running. Okay. And like I said before, this picture number 2 is what we've just set up. So here's our popsicle stick. our chromatography paper hanging down, and here's our solvent. And again, we're making sure that the paper doesn't touch the edge of the beaker, and we're also making sure that the solvent doesn't touch the little samples of the mixture that we've added on here. Okay. All right. I don't want to get too ahead of myself here. Let me go ahead and put the next page for you. You can start kind of digesting this. Okay. But what we should be seeing happening in our beaker at this point is we should see that there will probably be a stain left from where our sample mixture was. But as that mixture is separated into its different components, the different compounds, we'll start to see kind of a streaking look, and then we may even see a couple of different edges of the pigments. Or in some cases, you may even see different colors of pigments starting to separate out. It's a great chance, if you're doing this with lower grades, to talk about how colors separate, so primary versus secondary colors. A lot of times we spend a lot of time teaching kids to put red and blue together and make purple. This is a chance for you to be able to separate those and ask the kids, does red and blue really make purple? And so with our experiment, since we're doing green, our prior knowledge tells us that we should be seeing at least two colors in here. We should be seeing yellow and blue. So let's zoom in and see how we're doing on this chromatograph. And it looks like we're about 1 centimeter from where we need to be to stop. So the reason why the water is moving through the paper, we said, is capillary action. But you may be wondering why these compounds are moving, and unfortunately I can't zoom in anymore, but if I were to zoom in, you would see a little bit of color separation. So for example, the name brand marker, it's over here on the left, you'll see a separation with some yellow at the bottom, and then blue is on top of the yellow. It's actually risen above the yellow, which means it's moved farther. So how does that happen? Why do some compounds move faster or farther than others? Well, there's a lot of reasons, especially with paper chromatography. It's not super cut and dried. But a general rule of thumb is that it's going to be dealing with the polarity of the solvent and the compounds and how dissolvable those solutes are within the solvent we're using. In other words, how well does that yellow pigment and how well does that blue pigment dissolve in the water that's traveling up through the paper? So just generally speaking, we can say that if a solute is more soluble in the solvent, then it's going to move faster, which means it's going to move farther. If the solute is less soluble in the solvent that we're using, it's going to move a little bit slower, which means it doesn't go as far through the paper. So for example, here with our yellow being on the bottom and our blue being on the top, that tells us that yellow is going to be the less soluble of the two colors, the two pigments, and blue is going to be the more soluble. All right, so it looks like we're about 2 centimeters from the top, so I'm going to take my cover off. I'm just going to set that to the side for now. And we're going to lift our chromatography paper up and out of the water. And then, whoops. You probably shouldn't do that. You want to try not to drop that back in there. So I'm just going to lay this on a tray to dry out. All right. And the nice thing about this experiment is that you can reuse the water over and over and over again for this experiment. You don't have to change it out for each round unless somebody has dissolved all of their mixture into it by accident because they touched that those little dots into the water. But as long as the water is still fairly clear, it can be reused. All right. So again, I don't know that I can zoom in anymore. But what we're seeing on this sheet is that for the name brand washable school marker, we have some yellow pigment that's separated on the bottom. There's some green streaking here, which I'm guessing is because some of the blue pigment just hasn't gone far enough up on the paper, but then we have some blue compounds. So that tells us that the name brand green marker is a combination of yellow and blue pigments. Then our second off-brand marker also has some yellow and then some blue around the top. And this one's a little bit messier. That could be because it's different compounds, different pigments that are used. It could also just be a flaw in the paper. We're not using super high quality science chromatography paper here. We're just using coffee filters. So that would be my guess with that weird spread. But we are seeing that it's a combination of yellow and blue pigments. And then last but not least, we have our chart marker. And that's down here at the bottom. And this one's really interesting because we saw on the two school markers that yellow was on the bottom and blue was on the top. But this one, the blue pigment actually stayed towards the bottom, so it was less soluble. And the yellow pigment is way up here, so it's actually more soluble. So a little bit different formulation for this chart marker. So it's kind of interesting. And you could compare this to permanent marker. You could compare this to pens that are green. I'm a huge fan of those flare pens. And so you could definitely put some green flare pen on there and see what happens with that as well. All right, so now that we've seen that, we've done a visual comparison, right? So we just kind of held this up and we went, well, yellow, blue, and we compared them visually to each other. But what if you were looking at unknowns? How would you compare those? For example, let's say that I needed to name a certain pigment or I needed to figure out exactly what pigment that was. How do I determine that? Well, for each pigment, or each compound that exists, when it's dissolved in a certain solvent, and it's always run in a chromatogram on the exact same kind of chromatography paper, it has what's called an RF value. And that RF value is constant, again, like I said, as long as the solvent and the paper don't change. So that's what I have up here on the board right now. I'm just going to introduce you to this idea. This is not something I would do with the younger grades, but it's definitely something that you can introduce with older students. We see this happening, the same idea happening in DNA electrophoresis gels, in that we can run different proteins or various other substances on those gels, and we can look at certain markers, and whenever we see something end up at a marker, we have a pretty good idea of what the name of that compound is. Well, it's the same idea here. So when we're looking at RF values, the way this works, and you're going to have to kind of use your imagination here that this is tiny and normal size, but what I would do is I have to look at the distance that the solute traveled over the distance the solvent traveled, and that gives me that RF constant for filter paper in water on these markers. So what I would do is I would measure in centimeters, of course, the distance from that origin pencil line to wherever the solvent front ended. Now, with water, the solvent front hangs around for quite a while, so I can still see where the solvent front is. But for some of your more volatile solutions like acetone or petroleum ether, you're going to have to actually put a pencil mark where that solvent front was when you took it out of the solvent, because it will evaporate so quickly you won't be able to tell. All right, but for mine, I can still see it, so that's always helpful. So here on my picture, I've marked the solvent front. So I'm going to measure from the origin to the solvent front, and whatever that number happens to be, that becomes my denominator. So in this case, again, using our imaginations, the distance that my solvent traveled from here to here is 10 centimeters. I know, it takes a lot of imagination for that one. Then, in order to determine the RF value, we have to measure to the top of the solute front. So I'm going to measure to this bottom pigment here because I want to know what that is. So again, in centimeters, I'm going to measure from my origin line to the top of that solvent point. Now, as you saw on mine, and as I've drawn up here on the board, they're kind of bumpy. So it's not like a pretty straight line. It'd be nice if it was, but you know, science is messy, right? So as long as you measure to the same point along the same line on all of these, that's the important thing. So for this first one, I would measure here. Then if I wanted to measure the second, I wouldn't move my ruler at all. I'd just make the same measurement again without moving. So when I measure from my origin to the top of this particular solute, it measures at 5 centimeters. Okay. Now at this point, we could reduce our fraction just to make the math a little bit prettier there. Okay. And let me zoom in so you can see what I'm doing just a little bit better here. Sorry, I didn't realize I had zoomed quite so far out. Okay, sorry about that. So coming back over here to our chart, then we want to put our RF value into a decimal point. So we would just divide 1 by 5, which gives us an RF value of 0.2. There's no label, centimeters cancel out, and RF has no label, so RF is just 0.2. We could do the same thing for our second pigment, get that RF value. But basically what this would do then is I could go back and see if there's a chart that gives me RF values of marker pigments in water, and whichever one of those is 0.2 would give me the name of that pigment. Again, this is a little bit more of an advanced piece of chromatography, but it's a good way to identify specific compounds. And when we talk in just a minute about the different real-world uses of chromatography, this is one of the things that they do when they're trying to determine an unknown substance. All right. Hi, Mitzi. Thanks for joining us. Sorry, I just saw your comment. Glad you're here. All right, so that's how we do chromatography. So why do we care? What do we use this for in real life, right? It's great, it's pretty. We get all these pretty rainbow colors, and here's one I did earlier that's like super rainbowy. You know, it's always fun stuff. This one's not quite as good, but still some pretty colors. But what's the point? Why do I need to know this? If I'm a kid at this point, I'm going, wow, that's so cool. So what, right? Well, there are some real-world uses for this. Let's zoom in there. So, in real life, what do we use all this for? Well, one of the uses is that we can do chromatography on medications, and we can determine how pure those medications are. So when a company develops a medicine, or if they're producing a medicine that they already make, they can take samples of that off the line, go in and do chromatography on that, and the chromatogram will tell them if there's anything dissolved in that medication that shouldn't be in there. So they would know from their original development what dots should be where. In other words, their version of what color should be where. And then if there's something extra that shows up, they know something's wrong with that batch. We also see it used to identify pesticides that exist in fruit and vegetable skins. So when we run a chromatograph of plant skins, you do get certain pigments, certain colors, and then you can look to see if a pesticide shows up. We also can run chromatography where amino acids are separated out. And so again, if something shows up in that that's not supposed to be there. Substance identification in blood. Some of those banned substances, for example, this year you saw at the Winter Olympics that the Russian athletes were competing under the Olympic flag because Russia had been banned. And so one of the ways that they can identify those substances that might exist in the blood of an athlete is by doing a blood sample and then they can go in, do a chromatograph, and see what is separated, and they can determine, using the RF values, what those substances that exist in the blood are, and they can compare them back to a chart and see if they're legal substances or if they're not legal. We can also do this after someone has died to determine if there were any drugs or alcohol in that person's system as well. Arsenic, you always see those moments in history where people have been poisoned by arsenic, and this is one way that we can detect that, or we can even pull samples of food and drinks that exist in a crime scene, and we can determine what's been mixed in with those. We can use it to detect contaminants in water sources, so you can take a sample of water, run that out, and look at what's mixed in there. And then this is the one that I found most interesting, is that we can actually use chromatography to combat the Ebola virus. And so what we do is we can use the process to determine which antibodies create the biggest response to the Ebola virus. And while unfortunately we haven't come up with any drugs that are super great and have those perfect antibodies to destroy the Ebola virus, it has helped with developing some of the drugs that are starting to combat Ebola. So those are just some of our real life uses for chromatography. Awesome. Great question, Mitzi. There is a modified way that you could do this using water as a source. I'm going to have to do some more research on that. Right off the top of my head, I don't know what materials would be specifically required for that, but I do have a resource that I can look and see, and I'll post a link to that on Edmodo for you. So, hi, lady. Glad you're able to join us too. Awesome. Yeah, but that's a great question, Mitzi, about how to tie it into your unit on weather and rain. Which brings me to my next topic, which is how do you use this in your classroom? So making rainbows is fun. This is my high-tech PowerPoint here. So some ideas for your classroom, and I'm going to show you a couple of these in a second. So the first one is what you've seen already. It's the color separation, right? So how do we view primary colors versus secondary colors? And so, again, already demonstrated that. This one's getting a little drier, and so you can see the difference that we did earlier. So great for primary school. Another idea that you can do with the color separation is rather than separate it out on a little square like this, you can do it on the whole entire coffee filter. So here's a couple that I did earlier. Let me zoom in so you can see those better here. There we go. And so I ran these on the whole coffee filter. So how did I get this? Well, what I did was I started with a whole entire coffee filter like this, and I folded it into a pie shape. So fold it in half, fold it in half again, and one more time. Make some nice creases there. Ta-da. Then we unfold, and now I have a dot in the middle. So what I did was I drew a circle using just the bottom of a plastic cup. Make sure that's in the, there we go. Getting used to the view on this thing. So I used my pencil, I drew a circle all the way around the bottom of my plastic cup. Ta-da! That's the equivalent of drawing the line on the bottom of the square. Then once I had that circle, I grabbed a marker, and we'll use brown. Oh, let's do the off-brand brown. And then I basically just drew a big thick line of brown, or whatever color you pick, all the way around that circle. So here's one I did with blue earlier, and the blue marker was kind of starting to dry out, so I went over and over and over it again. Then I just put some water into the bottom of just a plastic cup. This is just a boring old plastic cup. There we go. Then I laid the filter on top, poked down in the middle, kind of messed with it. Let's try it this way. Now, ideally you'd want to let this dry before you do this. So like let the marker dry all the way. And then I just used a pencil to kind of hold the tip down in there. There we go. And then I just let the same thing happen. Now, I'm not covering this one because I am using water, so it's not as big a deal. There's not going to be evaporation happening as quickly as with other solvents. You could cover it though. But in just a minute, what you'll see you get is this really interesting design. So I'm going to slide this over to the side, because then if you wanted to turn this into a craft, and this is great for like, you know, your super, super younger grades, you can have your students do a couple of these and then kind of zigzag fold them. This is a very bad zigzag fold. So I'm going to fold that one. I'm going to add my purple one. And then you can grab just a fuzzy chenille stick, also known as a pipe cleaner. And then twist that on there. And you have chromatography butterflies. Another thing you could do if you're doing a unit on plants, I'm going to take my butterfly apart here, is you could do the chromatography first and then have your students during their unit on plants model the parts of a plant and create a three-dimensional model. And so they can twist up their chromatogram that's on the sheet to use as their petals for their flower, and they could trim them. And then you could have them put like the stamen and the pistil in here, so the male and female parts of the flower. You have your stem. You could have them model the leaves, model roots, and tie this into another unit as well. So they would not only be doing the doing the pigment separation, but then they would also be modeling for your botany unit. So a couple options there. All right, so that's the color separation. Another thing you can do is a modified CSI lab. So if your kids are really into that forensic piece, you basically do what we did with the 3 different colors of marker. And so you can have your students solve a crime, essentially. And this is what I used to do in my classroom. So what I would do, let me find a piece of paper here, is I would tell them that at the scene of some sort of incident, we found someone's signature. And so I've taken a small piece of that signature and I've put it onto the bottom of my chromatography paper. And then I run this for the entire class. And that becomes the mystery substance. And then they have to figure out which marker wrote that particular note. And so basically it boils down to the note was found. You have 3 suspects. Each one possesses a different blue marker. The kids have to figure out which blue marker wrote this. So you have the known with the mystery substance on it, and then the kids go through and create a chromatograph, kind of like we did earlier, and they have to figure out which one matches best. And because this runs so quick, it's a great example of how in science we do multiple trials. So the kids can actually do this, 3-4 times to confirm their results, and it's low cost for you. So just real great science process piece as well. I've also done the same thing using pens with my older students. And so, everybody has like a ton of different black pens. A lot of people don't realize that black ink is not solid black. So I would just gather up like a bunch of different black pens. Again, I'd write a message with one, we'd run that one, and then the kids would compare that to the suspect pens. So there's that one. Another option is to identify plant pigments. If you're going to run the plant pigments, it's a little bit different. You can look that one up online. It does not work with water. The plant pigments are not soluble in water, so you have to use a different solvent. And then another option, and Mitzi, this is the one that I was speaking to earlier on your question is an environmental engineering piece. And I'm pointing over here and you can't even see what I'm pointing to, right? But this environmental engineering piece where the students can work to develop a remediation process to remove contaminants from water. And again, I'll send the link out to this. This comes from another organization. But basically the students do the chromatography and they're looking at what's contaminated the water source, which might be an interesting tie-in to weather and rain, since we know that rain can pick up various things. For example, at Chernobyl, the rain that was dripping in through the sarcophagus sealant structure that they had created before the new one from 2016. But when rain dripped in through that sarcophagus, it was picking up those radiation particles and washing them away. We see this underground as well. Water picks up minerals that are in the water and takes those away. So that could be an interesting tie-in as well, and a great engineering design piece for the kids where they actually have to create the process themselves and test it out. Awesome. Yeah, and I'll send the plant version out as well. There's a couple ways to do it. I will say that the solvent is a little more volatile for that one. So that one has to be done with a little bit more caution. All right. Let's change gears for just a second, and I'll go ahead and pull this one out that we just made. So, there we go. So this is the one we did with the brown marker. And again, I know it's hard to see from your distance, so I know you're all going to go home and dig out the coffee filters tonight, right? Play with it. But you can see multiple colors, so it turns into a pretty rainbow. And that was brown. A lot of kids don't realize that brown has so many colors in it. So there we go. All right, so those are just a couple of uses for you in your classroom. If you guys have suggestions, I would absolutely love to see those. You're welcome to put those in the chat box or have that conversation later on Edmodo. But that's pretty much the conclusion for the chromatography piece of this. So I hope you got some interesting information about the chromatography, what it is, ways that we can use it, and then some applications that you could possibly do in your classroom. I don't know why I'm playing with clothespin. All right. So if you have any questions, again, please feel free to put those in the chat box there. I do want to take just a second and give you a little bonus activity. I know this is the time of year that sometimes it's hard to keep kids entertained as well as on task, right? They're getting squirrely for summer. So one of the games that I found online a few years back and my kids fell in love with, and I taught 9th through 12th grade, and my kids would play this for 30 minutes at a time and they never got bored. And the best part is it's the game that never ends. So if you haven't heard of this game is called Kaboom. And it's super easy. It's set up with some popsicle sticks, a Solo cup, and a marker. So what I do is I go through, and let me zoom in here so you can see. I color one side of one end of one side of the popsicle stick with a marker. So like I've just colored this one blue, and on the back there's no color. This serves two purposes. The first purpose this serves is that if I have two sets of these, so like here's two sets, I have a blue set and a pink set. So if they get mixed up, I can tell really quick. That goes with that set, this one goes with the blue. The other reason is that the color on one side indicates to my students that that's going to be the question side, the blank side's the answer side. So when the kids grab the stick during the game, they know, look at the color side first, and then when I flip it over and there's no color, that's the answer. So they want to look at the side with the mark first. So the way this works, you put your questions and answers onto some popsicle sticks. So here, this is just real simple. This one I've done multiplication. So I've just written like 4 times 1. And that's on the question side. And then on the back is the answer. And the way it works is that you have multiple questions. So like here's 4 times 5 is 20. And then every few sticks or so, 1 in 10, I always say at the minimum, you write kaboom on one of the sticks. And you can put 3-4 kabooms in a cup depending on what you on how many problems you have and how many kids are going to be playing. So the kids work in groups. They can work in 2s, 3s, 4s. I never really went over 4. I just felt like that was too big of a group. But the way this works, the first kid grabs the stick, pulls it out. 3 times 3. If they answer correctly, then they get to pick another stick. If they answer incorrectly, then the cup goes to the next person. So I'd be like, 3 times 3 is 9. Yay, me. I pull out the next stick. 4 times 5. 4 times 5 is 15. Oh, check my answer. Nope, that was wrong. So I pass it on to the next kid. If I get the answer right, I keep the stick. If I get the answer wrong, it goes back in the cup. The kids keep going around in a circle, drawing and answering, saving or putting back in, until one of the kids draws the kaboom stick. When they draw the kaboom stick, then they take all of their sticks and they go back in the cup. Now, what's so cool about this is I used to tell my kids that the kid with the most sticks at the end wins. The thing is that because of this kaboom stick, the game never ends. So as soon as somebody draws it, even if they have no sticks, they put the kaboom stick back in, the next student pulls the kaboom stick and all their sticks go back in the cup. So it's the never-ending game. So like I said, my kids would play for 30 minutes at a time and they're like, man, I'm getting so close to winning and I never do. And it's because they kept pulling that kaboom stick out. So just a quick way to review, and it's quick for you to make. I taught older grades, and so it was way too complicated for me to write on all my sticks. So what I used to do is I would print out labels. So again, pink is my question side. Whoops. So here's the question, and I just print these on Avery labels, and then I just trim the top and bottom of them so that it fits on the popsicle stick. So like, why are flower petals brightly colored on a plant? And then answer, they attract pollinators for reproduction. But by using the stickers, I could put larger questions on there and then have the kids give more complicated answers. It's a great test review game. It's a great way to keep them busy after they finish their work in class, a good sponge activity, and then, of course, the magical kaboom stick. So just wanted to share the idea of the kaboom game with you so that you would have that tool in your belt. These little guys too, they store really easily, so you can just like wrap them up with a rubber band, or you can just put them in a Ziploc bag. And I used to store all of my kaboom sticks in just one big box for the year, and I just had the baggies labeled with the unit. So it was like plant reproduction unit, there they are, or DNA unit, here they are. And it was real quick to make multiple sets so my whole class could play at one time. And then I always had sets just sitting in my sponge activity center. So just wanted to share that idea with you guys today. And I hope that you have found something in this workshop useful. I appreciate the excitement over here on the chat. So I'm glad that these are all things that you guys are going to be able to use. If you have any questions, of course, feel free to email. I'll hang out on the chat for just a couple more minutes. And you can always email us at teacherworkshops@perotmuseum.org. And if you are not currently part of our STEM Teacher Institute, we would love for you to join us for our upcoming workshops. And we post all of our public workshops at perotmuseum.org/professional development. You can check out what we have to offer there. If you are in the STEM Teacher Institute, then we're excited to see you in a couple weeks when we start our summer academies. And the last thing that we have to do today is make sure that you get your CPE hours. And so for that, I will post this as well in the chat, but we have a survey for you online. If you will fill out the survey at this short link and give us your name, email, do the reflective piece, then we'll send you a certificate for 2 CPE hours. And like I said, let me post that in the chat right now. And I'm sorry that's as close as I can zoom in there. But from my preview screen, it looks like it's pretty visible. So it's https://goo.gl/lowercase h, capital B, capital Q, capital C, the number 3, and then a lowercase p, as in popcorn. All right. And that link will stay active for at least 48 hours. So you'll have a chance to get that done, and then we can get those certificates sent to you. All right, so thanks again for joining us today for our live webinar. This is hopefully the first of many of these. Again, I'm Mary. I'm the Professional Development Manager here at the Pro Museum, and we're so glad that you joined us today and learned with us, and we hope to see you again soon. Bye, and have a great evening.